In today's video, we are going to do some detective work. The case involves a human metabolic disorder where gene B is not being expressed properly in the liver. The result of gene B not being expressed properly causes the body to be unable to break down substances. Some key points in this case are that gene B is expressed normally in the pancreas, and no mutations are found in the coding region of gene B in either tissue. This disorder may be a result of changes during gene regulation, since gene regulation takes place at multiple levels. This diagram describes the multiple levels of gene regulation. Now, let's get to the hypotheses. Our first hypothesis takes place prior to the formation of pre-mRNA. This hypothesis states that DNA methylation occurs in the liver cells, but not in the pancreas cells. DNA methylation is a process in which methyl groups are added to a particular DNA segment without changing the DNA sequence. These DNA methyltransferases tend to cause genes to be turned off, thus silencing them. In this case, it would silence gene B in the liver, thus not allowing it to be expressed. DNA methylation can be activated due to aging, environmental influences, and lifestyle factors, like excessive alcohol consumption, smoking, or poor dietary choices. The second hypothesis affects expression after the pre-mRNA formation. The reason for gene B to not be expressed properly in the liver is a result of RNA interference. RNA interference shuts off gene expression by using double-stranded RNA. Depending if siRNA or microRNA is present will determine what happens to the gene. When siRNA is present, it results in the cleavage and then degradation of mRNA. siRNA also causes transcriptional silencing. This alters chromatin structure by siRNAs binding to the chromatin, attracting enzymes that methylate the tails of histones, then causing the DNA to bind to the histone more tightly and decreasing transcription. All in all, interfering with gene B being properly expressed. When microRNAs are present, translation is inhibited. This occurs when microRNAs are paired with mRNAs, again, resulting in gene B not being expressed properly in the liver.